I'd like to also welcome all of you to the Dairy Research Cluster's first biannual symposium, Innovate 3. And of course, the three pros are promoting progress to the profit of the dairy sector and Canadians. This event provides an opportunity to hear about the latest progress resulting from the dairy farmers' research investments and the Dairy Research Cluster and associated research initiatives. Experts from each of our priority areas of research will present their latest findings and how the outcomes can contribute to improving the way we care for our animals, enhance our management practices to be more sustainable, and gain new insights on how our management practices and how our dairy products benefit Canadians' health. That is why we refer to our research program as Dairy Research for a Healthy World. It includes our animals, our environment, and our consumers. Isabel will provide an overview of how our current activities under the program in her presentation. Dairy Farmers of Canada has proudly been investing in research for more than 20 years now. Through these investments and a sound process used to identify the best scientific projects to get the job done, the results have led to practical solutions to the issues facing us on our farms and across the sector, and support our efforts to make the best products to feed Canadians. So over 20 years, we've worked with a strong community of established dairy scientists that are well respected in Canada and internationally, many of which you will hear from today. Our program has also provided opportunities to train students in dairy sciences and support the next generation of dairy scientists. You'll have a chance to see some of their work today in this video challenge we put to the students, asking them to present their research in 180 seconds. And I'm sure that's quite a feat. I encourage you to watch the videos right here after lunch, and we will ask you to vote on the best one. Finalists are here with us today, and DFC is happy to provide a $500 prize to the winner. And I'll be announcing the winner at the end of the day. Our research is designed to help us improve, to be more sustainable, develop new technologies, and innovate. Science-based practices in dairy are not new to us. Take our animal care assessment program in ProAction, for example. From the inception of the Code of Practice for the care and handling of farm animals, for dairy cattle, to the ProAction on-farm assessments we're rolling out today, our farm initiatives are backed by the best body of scientific evidence we have available to support our commitment to Canadians. And that is that we are meeting the highest standards for milk production, that we will work to ensure milk quality and safety, and to continually improve animal health and welfare, as well as environmental stewardship on the farms, on our farms. With 20 years of experience in this portfolio, we know we need to keep innovating and identifying issues on farms to be even better businesses and contributors to Canadian society. In 2015, we asked farmers and dairy stakeholders to have their say in Canadian dairy research. The response was positive, and the conversations in farm meetings across the country were very thoughtful. Emmy will tell you more about these results in the next presentation. But what really strikes me here is that we all want to keep on improving. We work with our animals every day, and we know firsthand that a healthy, comfortable cow is also a productive cow. The survey prompted discussions among farm leaders, and I'm pleased to announce that following our meetings in this past week, DFC will be launching the development of a national strategy for dairy research and knowledge transfer, led by a new national body under DFC called the Dairy Research Council of Canada. We will be developing a coordinated and a collaborative initiative with our provincial members to invest our respective dollars in the research that will provide the most value to farmers, ensure that research findings are transferred efficiently and effectively to farmers, and continue to investigate and promote the benefits of dairy products to Canadians' health. Emmy will also report on the details of this initiative next. And if this is certainly something that we're doing in many other areas as well, where we're trying to have better coordination of what's happening provincially and coordinate that with what's happening nationally, which should certainly improve the efficiencies of our investment. In closing, I want to thank our investment partners in cluster and associated research, Agriculture and Agri-Food Canada, the Canadian Dairy Commission, the Canadian Dairy Network, and the Natural Sciences and Engineering Research Council. DFC's cluster investments are matched on a ratio of 1:3 for a total portfolio envelope valued at $18.9 million, allowing us to take major strides in advancing and innovating in Canadian dairy. And recently, Agriculture and Agri-Food Canada has approved investments of an additional $1.8 million in 4 project areas. The first one is increasing the energy in forages fed to cows. The second, investigate the nutritional synergy between dairy products and other food nutrients. And third, study opportunities to reduce sodium in cheeses. And finally, to identify the best products to provide sources of vitamin B12 from the cow to the consumer. And so we have greatly appreciate and continue to appreciate the support that we receive in terms of funding, which puts together a fund which is very significant, and it allows us to do a lot of very important work in these areas. And so with that, I would again like to welcome everybody here. I thank you for your attention, and I hope that we will all have a very productive day today during our conference. Thank you.
Thanks very much. Big announcements, good news, very exciting future for dairy research. So I invite Isabelle and Amy to present and talk a little bit more about the details of the upcoming events.
Alors, bon matin tout le monde.
Good morning, everyone. I will start off in French, and midway I'll move on to English. I will, first of all, talk about the current situation with regard to research and knowledge transfer at DFC, and specifically within the cluster, which is our main program. And then, Amy will talk to us about our plans for the future, and will tell us more about the next stages of our research programs. So, for the last year, research on dairy farming, dairy production, and human nutrition are within the same department. But there have been some changes within the DFC and now research in dairy production and human nutrition that have now come under the same department. There's myself, who is Director Manager of Nutrition, Maria Kalurgis, National Program Manager, responsible for scientific affairs in relation with nutrition. Maria is in the room. If you haven't met her, please go and talk to her. Amy Desilets, Assistant Director, Dairy Production Research, and she manages the Dairy Research Cluster. Annick L'Espérance is the Research Coordinator for the Dairy Research Cluster, and she is also in the room with us. And Shelley Crabtree, that we met earlier, who is our research communications specialist. And we are very enthusiastic about working together and very keen on this change, because it facilitates the interactions and the cohesion between and the integrations of the research activities in the areas of milk production and dairy nutrition. For those of you who are less familiar with our organization, let me tell you who we are as Dairy Farmers of Canada. We are the voice of Canadian dairy farmers across Canada. We are an organization that is run for farmers, by farmers, and we are Dairy Farmers of Canada. Dairy Farmers of Canada is the national policy, lobbying, and promotional organization representing Canada's farmers living on 11,962 dairy farms. And we strive to create stable conditions for the dairy industry. We maintain policies to foster the viability of Canadian dairy farms and promote dairy products and their health benefits. DFC has been financing research for the last 30 years, and today they invest about $2 million annually in dairy production and human nutrition research. Thanks to our various partnerships, the value generated by our research effort last year through DFC's financing was to the tune of $6.6 million. And this is without counting all the research initiative and knowledge transfer initiatives undertaken at the provincial level. So it is major. This funding is directed annually to address farmers' priorities for research in milk production and in human nutrition and health, which are closely linked to providing science-based evidence and farm practices. And these are proaction, productivity, efficiency of farms, sustainability, and human nutrition. Under the theme Dairy Research for a Healthy World, it is the tagline for DFC's research program, and we aim at improving the health of our cows, our environment, and our consumers. DFC leverages its investments by partnering with governments and other organizations in major funding programs, including Agri-Innovation, the Agri-Innovation Program from Agriculture and Agri-Food Canada for the Dairy Research Cluster and the Organic Science Cluster, but we also have an annual competition in human nutrition from Dairy Farmers of Canada. And we also have the grants for NSERC, as well as the Dairy Gen Committee of the Canadian Dairy Network Open Competition. We also have the DFC's Production Expert Scientific Advisory Committee annual competition, which was temporarily suspended because of a lack of funds. And we hope we can restart this program in a very near future. I'll skip this slide because David did mention it. Did mention this. So
if you need to put your headphones, give you a few seconds. So DFC's research program is currently supporting a total of 45 research activities involving 144 scientists from 13 Canadian universities and 10 federal research centers across the country. So that's very impressive. In addition, more than 130 graduate students and postdoctoral fellows will be trained over the next 5 years. The program also supports an extensive communication and knowledge transfer plan, which Shelley will be talking about in a couple of minutes. So this brings me to our communication and knowledge and technology transfer activities that are part of our research program. We have several activities to ensure the transfer of knowledge and technology from the studies that we fund. There is our research portal. The Canadian Dairy Research Portal contains information on over 2,000 Canadian dairy researchers and Canadian and international collaborators, and over 520 projects submitted to DFC for funding since 1996. Our users on the site doubled last year, with average monthly visits of about 500 and over 20,000 page views viewed over the year. We also have the Dairy Nutrition website, which is the DFC website dedicated to health professionals. We have the Dairy Research blog, where we publish about 2 to 5 stories monthly, which are sent to 150 subscribers. We also use the YouTube channel. We have 25 videos on the Dairy Research Cluster channel with content on animal welfare, the life cycle analysis, nutrition research, results, and more. And finally, we are also active on social media weekly, especially Twitter, and have almost 1,100 followers. In addition to the promotional and informational material printed for the cluster and traditional media, we also write about 50 to 60 articles a year for extension purposes in collaboration with researchers or on our own that we distribute through various columns in numerous publications, and you have a few examples of those on the screen. And we also publish a research update via an electronic newsletter. Lastly, we attend or present at 8 to 12 farmer conferences throughout the year with our research booth, or we provide support for the development of material to be handed out during those meetings. And we collaborate on research results to facilitate and coordinate the production of videos and webinars. We also have several tools at our disposal as part of our nutrition communications program to convey favorable results, such as the NE newsletter for health professionals, our website, and our annual symposium for health professionals as well. So our research process includes a rigorous cycle that can be divided into 4 steps. The first one is the identification of issues and the establishment of priorities. And then we proceed with a call for proposals that is followed by a 2-stage selection process based on relevance to dairy farmers, an assessment of the scientific value, and a peer review. The third step is the submission of an application to a funding organization and a review process as well there. And finally, when the project is accepted, it goes through an ethics committee from the participating institutions and the signing of agreements between both various parties. Given our success and the appreciation of the program, we really hope that the government will renew the cluster initiative or offer another similar research program when the cluster program, Cluster 2, ends. We are very optimistic, though. We have to believe. So in anticipation of that, we have already started to prepare for a post going forward too. And I'll let Amy tell us all about it.
Thank you, Isabelle. I will do my presentation in French. Donc, Isabelle vous a un peu...
So Isabelle gave you a description of the situation where we're at in terms of dairy research and knowledge transfer. So before I tell you what's on the program for 2016, allow me to go back to the campaign we launched, Have Your Say, on dairy research that we conducted throughout 2015. Through an online survey, we asked dairy farmers and other stakeholders of industry to help us shape dairy research priorities. So we invited dairy farmers to have their say by identifying a farm issue to be solved by research, by asking a scientist a question, or by identifying research priorities for the future. Through these tools, we participated in 15 conferences and meetings across the country. When I say we, it involves Shelley, who's here. So Shelley has crisscrossed the country to meet producers everywhere and to engage farmers and other decision makers in helping shape dairy research priorities. The respondents represented various sectors of our industry, those who were involved in the survey, 71% were dairy farmers, 61% were dairy farmers, and we're very satisfied with this, very happy with this, because they are the ones who are our main target audience. Now, the responses obtained were very varied and often based on the individual concerns of the participants. This week, as part of the annual policy conference, we consulted our delegates and producers to determine what are the answers or responses that were the most important to ensure the sustainability of our industry in the future. All these answers will be compiled and will allow us to shape the future. They'll be a basis for discussion, and they will allow us to define our priority research, and that have been going on since 2011. Now we are kickstarting again a new cycle. But DFC this time around want to go further. We do not simply want to define research priorities. We want to develop with the various provincial organizations a national strategy for research and knowledge transfer in dairy production and in human nutrition. So this strategy would first of all reflect, as Isabelle said, our new structure. Dairy research and human nutrition now really work hand in hand. For you from the outside, it wasn't probably apparent, but for us from the inside, it's a major change in the way we do things. And we are really convinced that it is for the best. Other initiatives will be undertaken by DFC. Now, at the national level, there's an effort that's made to involve more concretely our provincial member organizations at the level of issues that might be led by DFC, but I love the expression of one of my colleagues, we want to put the members of our provincial organizations in the driver's seat with us. So this is part and parcel of this new momentum. Now, to go back to this strategy on research and knowledge transfer, will facilitate, ensure communication, improve coordination, maximize collaboration among provincial and national producer organizations in planning and delivering dairy research and transfer programs across the country. All this with a view of avoiding duplication of efforts in research and optimizing investments. It goes without saying that the strategy must ensure that dairy research and knowledge transfer activities support farm efficiency and sustainability, that they stimulate innovation, that they enhance producer-led programs such as ProAction, and that they provide credible and compelling findings about the benefits of dairy products for Canadians' nutrition and health. So Dairy Farmers of Canada will establish a new governance structure and create the Canadian Dairy Research Council. And reporting to DFC's Board of Directors with a first mandate to develop a national strategy for dairy research and transfer in Canada. Now, the Canadian Dairy Research Council will be made up of staff of each province, and DFC board representatives. Since we are at the beginning of the process, I should have mentioned that this committee will be, how can I say this, will receive recommendations, and we could have included the technical research committees that already exist. Experts in nutrition and in dairy production will be part of the initiative as well. Once things get underway, we also hope that we can strike a committee that would be in charge of transfer, but we're less advanced on that side of things. Now, action plan and timeline as approved by the Board of Directors of DFC for the development of the strategy. First of all, creation of the CDRC, which should be done very shortly. It's the Canadian Dairy Research Council. This was discussed this very week. In April 2016, we will facilitate a consultation session with industry stakeholders to build, as it were, what we need and what the strategy will include. The goal is to present the national strategy for dairy research to the DFC General Council for adoption at the AGM in July of this year, and to present the national strategy for knowledge transfer to the DFC General Council for adoption at the next annual policy conference, that is February 2017. As you might have noted, our action plan is in line with what we are anticipating in preparation for post-GF2. We've had meetings earlier this week from people from Agriculture Canada, and it seems that the renewal of a comparable program to the cluster program is likely, is probable. So we see this very positively, and we will stop the process to prepare ahead of time, as we had done as part for the first cluster. Now, according to this action plan, we will make a call for proposals, and by the end of 2016, beginning of 2017, it will be done. We will keep you, of course, as researchers, well abreast of this new call for proposals. Thank you very much for your attention.
Good morning, everyone. Thank you very much for the invitation. I'm delighted to be here to talk to you about dairy products and cardiovascular health in the name of my superiors. Cardiovascular health, well, it's a very fascinating topic. Research in this area is aimed at a reduction in the consumption of saturated fatty acids. We want to reduce bad cholesterol in order to reduce the risk of cardiovascular disease. Dairy products, well, there are recommendations suggesting that people drink 1 or 2% milk, as well as fat-reduced dairy products such as cheese and yogurt, low fat. It's the same message that is being delivered in the US at present. However, if you look through scientific literature, you'll see studies that say that dairy products enhance health and reduce cardiovascular issues. There are several studies that show that dairy products have no impact on cardiovascular health. So the question we have might seem simple, but it is complex. Is dairy consumption beneficial, neutral, or detrimental with regard to cardiovascular health? We attempted to have an overall picture in order to answer this question, and it was a two-step process. First of all, a systematic assessment of epidemiological evidence. We sought out all of the meta-analyses of prospective cohort studies. This is the highest quality analysis that we can use in such an exercise. So we looked for all of these analyses that established a link between the risks for blood pressure issues, diabetes, etc. We also looked at clinical evidence in all of the controlled trials and random trials. We looked at all of the results to see what the link between the consumption of dairy products and bodily function. This study is being revised for publication in Advances in Nutrition. There are 19 analyses dealing with 120 epidemiological studies. This is a very broad compilation. As you can see in this table, several conclusions can be drawn from this research. As you can see, nowhere is the consumption of dairy products in any form is it linked to a negative impact on cardiovascular health. Secondly, when we look at this table line by line, you can see that the total consumption of dairy products is linked to a reduction of risks for hypertension, metabolic syndrome, type 2 diabetes, and stroke. When you look at the consumption of high-fat dairy products, here again we see that consumers of high-fat dairy products aren't at more of a risk of having a stroke, which is reassuring. Low-fat consumption seems to offer some benefits with regard to the risk of stroke and hypertension and type 2 diabetes. An important point when you compare lines 1 and 2, high-fat consumption and low-fat consumption. In fact, these two lines can't be compared one to the other. It seems that there is an advantage to consuming low-fat dairy products, but there's no study that really compared the impact if you substitute high-fat products over low-fat products. When you look at the consumption of milk, which is a low-fat product, 0 to 3% on average, there are benefits for cardiovascular health with regard to hypertension and metabolic syndrome. Cheese is much richer in saturated fat, but there are benefits with regard to stroke and type 2 diabetes. And this is the case because the saturated fat in dairy is perhaps not an issue. This study wasn't conclusive in that area. We can also see that we've written uncertain in several columns. This is where in those studies there were either equal benefits or equal disadvantages. There's still a lot of work to be done when we're talking about cardiovascular issues or metabolic syndrome. I move on now to clinical evidence. This is another manuscript under review. So we reviewed 100 clinical trials. Here again, they assessed the consumption of all sorts of categories of dairy products, the total consumption of milk, cheese, no dairy products at all, etc. It depended. We talked about blood lipids, everything involving cholesterol, good, bad, and the various ratios used to assess cardiovascular health, inflammation, insulin resistance, blood pressure, endothelial function, and hemostatic function. These risk factors, some of them are more conventional, but we decided to look at them overall to have a comprehensive picture of the impact of the consumption of dairy products on these various risk factors and cardiometabolic function. Our conclusions were similar to what we found in the previous set of studies. No impact on blood lipids, inflammation, insulin resistance, blood pressure, etc. So I come back to my first question. Is dairy consumption beneficial, neutral, or detrimental to cardiovascular health? There's no high-quality evidence suggesting that dairy products consumption is detrimentally associated with cardiovascular health. But there is high-quality evidence, both clinical and epidemiological, that shows that dairy products consumption is neutral or beneficial for cardiovascular health. Thirdly, actual available evidence supports the inclusion of dairy products in a healthy diet. and it is recommended to have low-fat dairy integrated as part of healthy eating guidelines as opposed to regular or high-fat dairy. But this is not supported, this recommendation, by actual available evidence. And this is very important if we want scientifically based information. For the future, with Drs. Lamarche and Couture, we're going to be studying the following question. Does the dairy matrix food form influence the impact of dairy products on cardiovascular health? In the case of cheese, is the impact different because it's solid, and in butter we only have saturated fats? There are 3 clinical studies underway that are going to begin soon, that they're planned for, with support from the cluster. The first one aims at comparing a diet rich in cheese versus a diet rich in butter, its impact on good cholesterol and other risk factors. So we feed our participants over several weeks with a specific diet, rich in cheese, butter, low fat, PUFAs, MUFAs, and it's very complex. But this will allow us to determine if cheese presents an advantage versus butter, and if a high cheese diet offers the same benefits for health as other types of diets recognized as being healthy for cardiovascular health. Other clinical studies, we're going to compare different types of cheese. More solid cheese versus cream cheese. Does it have the same impact consumed in one meal on postprandial lipemia? And we want to compare cheese enriched in GABA, its benefits for cardiovascular health, versus milk. And as you saw earlier, milk seems to offer a lot of benefits for blood pressure. So, can we determine if there's a similar effect with cheese versus milk? All of these projects required the cooperation of about 120 researchers with participants throughout Canada. There are more than 16 students working on these projects as well. So there's an awful lot of activity that's been generated in INAF with Drs. Lamarche and Couture. Thank you very much. Thank you to everyone. And I am available if you have any questions. Thank you very much. We'll keep the questions for the end. In that way, we'll have an exchange questions and answers with our panelists. The next presentation, thank you again. The next panelist.
He's an associate professor in the Department of Nutritional Sciences at the University of Toronto and an associate scientist at the Leadership Sinai Center for Diabetes at Mount Sinai Hospital. His research focuses on the nutritional and metabolic epidemiology of type 2 diabetes and its underlying physiological disorders in high-risk populations. So I welcome Dr. Hanley.
Well, thank you, Shelley, for the kind introduction, and I'd like to thank everyone for the invitation to come and present some research to you today. So here's the outline of my talk. I'm really going to be focusing from a broad perspective on type 2 diabetes and the role that dairy products may play in the development of this condition. And so we'll take a broad look at the current literature in terms of what that's telling us today about the state of this research. I'll tell you a little bit about the work that's going on in my group, specifically in the area of circulating fatty acids and the impact on disorders that underlie diabetes. And then we'll take a look at the end at some potential future directions and conclusions. So it's well known to this group that the consumption of dairy is below recommendations. These are data from the Canadian Community Health Survey. The graph is showing you the proportion of underconsumption per recommendations for different age and gender groups. And so the blue bars are males, the red bars are females, and you can see that there is underconsumption across the board. It's particularly an issue for teenage girls and older individuals. Other data that sort of tells a similar story, these are data from the United States Department of Agriculture showing the decline in beverage milk consumption over time. For total beverage milk as well as whole milk, there was a slight increase over a period of time for low-fat milk, but that has leveled off. So at the same time, we see diabetes prevalence going in the opposite direction. And so this is a map produced by the International Diabetes Federation, which is the global body that advocates for diabetes research and care. Canada just hosted their international scientific meeting in Vancouver in December. I know some of the dairy farmers folks were at that meeting. And what we're looking at on this map are absolute numbers of people with diabetes, with the darker colors representing greater numbers of people with diabetes. And you can see, as we can imagine, the United States has a lot of people with diabetes. But what you're also noticing from this map is that this is really a global issue, right? We have diabetes touching everywhere in the world. And particularly we see this is a problem becoming an issue for developing countries, China and India. And if you can imagine the impact on those economies as the diabetes epidemic advances in those countries with all the devastating complications that come with diabetes, this is going to be a real problem. So the take-home message here, diabetes touches most places in the world. A point of clarification, most of this is type 2 diabetes, what we used to call adult onset diabetes, or what we used to call non-insulin dependent diabetes. This represents about 90% of the diabetes burden globally. There are some hotspots in the world for type 1 diabetes, and so Finland, my wife's home country, is a place where type 1 diabetes is particularly a problem. But really, when we look globally, when we look at the emergence of diabetes, it's mostly type 2. So the International Diabetes Federation estimates that there are approximately 400 million people living with diabetes today, and that by 2035, that number is going to jump to 600 million people. So a simple question, maybe a naive question, is we see dairy consumption and type 2 diabetes trending in opposite directions. Is there a connection there? So how do we go about studying that as scientists? Well, there are, and this is, for the scientists in the room, this is going to be a very crude simplification of how we do science, but there are two general categories, if you will, of scientific inquiry. Basic or mechanistic studies. These types of studies tend to happen in a laboratory using cell cultures or animal models. And then there are human studies, which include observational studies and randomized controlled trials. I'm going to tell you about the last two in the next slide. Jean-Philippe, in his presentation a moment ago, touched on these different designs as well. So I guess what I'll also encourage you to take away from meetings like this is that people will tell you, even scientists, some of my beloved scientific colleagues will try to tell you that some studies are better than others and that we could maybe ignore some lines of inquiry in favor of others. I don't personally believe that. I think all of those designs are necessary. Science comes to an answer by assembling little pieces of a puzzle. And those pieces have to come from animal studies, they have to come from trials, they have to come from observational studies. So all of it is important. So observational studies are basically defined by the idea that there is no intervention. People, participants, are measured very carefully, blood samples are taken, measurements are taken, and people are followed over time to see whether or not an outcome occurs. In my case, diabetes. Jean-Philippe was interested in cardiovascular disease. These studies tend to be larger and more long-term than trials, but they are limited, and I'll get to that in a moment. Randomized trials, on the other hand, the exposure is controlled by the random allocation of that treatment to an intervention or a control group. And for that reason, the evidence that comes from trials tends to be held in high regard because you are controlling for that intervention and you're taking away the biggest problem that can occur in observational studies, namely the idea that confounding can occur. So one of the things that happens to me when I have Thanksgiving dinner at my house, my non-scientific relatives say, there's so many studies, they don't always agree, we hear one thing one day, we hear one thing another day, how do we put it all together? Well, Jean-Philippe did a very nice job at telling you about meta-analysis, and in fact, the papers that he was talking about, these are going to be very important papers because they're reviews of meta-analyses, and so I look forward to seeing those when they come out. I'm just going to take you back a little bit to tell you about what a meta-analysis is. So this is a statistical approach to combine the results from multiple studies. The other thing to take away from a meeting like this is it's never about a single study. We hear news reports, and I'm not trying to toss reporters under the bus here because they have jobs to do. And in fact, some of this excitement about single studies comes from the media offices at our universities. It comes from our own scientists themselves. Meta-analysis allows us to take the collectivity of the evidence that's out there, and Jean-Philippe told you a little about what they're doing with cardiovascular health, and combine it all using statistical methods to see where we are today using all of the evidence. Brings many advantages, increasing statistical power, precision for subgroups of interest. The caveat here is that the meta-analysis relies on the quality of the studies that goes into it. So garbage in, garbage out, which is a problem with any kind of review like this. So what do the meta-analyses of observational studies tell us about total dairy intake and risk of type 2 diabetes? So don't be alarmed if you're not used to looking at meta-analysis results. We're going to walk through this. This is the main finding that you would see in a meta-analysis paper. If you're a fashion photographer, this is the money shot, okay? So on the right side of that slide, you see names and dates. These are the identities of individual studies that have gone into the meta-analysis. On the right side, you see numbers followed by numbers in brackets. And so the first number is the relative risk. If that number is above 1, it suggests to us that the exposure, in this case total dairy, is associated with an increased risk of the outcome, in this case diabetes. If the number is below 1, then we have the suggestion that the exposure is related to lower risk. The numbers in brackets are measures of precision. How sure are we about that result? In the middle, those boxes and whiskers are the graphical representation of those numbers. The bigger the box means that that was a more heavily weighted study, usually because it's a larger study, and therefore the whiskers tend to disappear on those big boxes. In the yellow highlight at the bottom, that is everything taken together. And so this meta-analysis suggests to us that total dairy intake was related to a lower risk of incident type 2 diabetes or new onset type 2 diabetes. Now, one of the nice things about Jean-Philippe's work is that there are many meta-analyses out there, and they're going to review them together. And so you can see on this next slide, we have other meta-analyses of total dairy intake and type 2 diabetes risk. They tend to tell a similar story for the most part. The two at the top are other meta-analyses that suggest, again, a lower risk of type 2 diabetes with the intake of total dairy. A more recent meta-analysis was more neutral, showing no significant effect either way, but this was heavily weighted by 3 new cohort studies that had recently been published that gave neutral results as well. So just to summarize this section, what do we know about meta-analyses from observational studies of total dairy intake and type 2 diabetes risk? Well, the message so far is that there is an inverse or protective relationship there. The most recent meta-analysis was neutral, although that was heavily weighted by 3 large cohorts that are highly selected in some ways. And I'll tell you, if you want to come and talk to me after, I can tell you about that. It's a little bit of a technical detail. What about individual dairy products? Milk, cheese, yogurt, etc. Jean-Philippe touched on this as well very nicely. Findings have been a little bit more mixed. We see stronger protective relationships for yogurt and low-fat dairy in some, but not all meta-analyses. This is just an example from one. We don't have to go into the detail here because this is a very complex slide. But here we see cheese and low-fat dairy in this particular meta-analysis were related to significantly lower risk of incident diabetes with neutral results for the other subtypes. In contrast, that latest meta-analysis that was overall neutral showed a protective relationship or an inverse relationship with yogurt. I only have one slide about trials, and in fact, Jean-Philippe did a nice job presenting where we are in this evidence. A lot of the results have not looked at diabetes as an outcome per se, but have looked at risk factors that can lead to diabetes. And what we generally are seeing thus far is that we don't see any significant changes for the supplementation of dairy in trials on waist circumference, glucose, insulin, blood lipids. There may be some modest evidence for increases in weight, but again, largely the evidence has been neutral. And what I would say is that we have relatively few studies in this area, and there's a wide variety of methodological issues: the duration of the trial, the type of dairy product that's being used, the outcomes that are being measured. All right, so I just want to talk a little bit about my own program right now. And there are two important concepts, conceptual things that drive a lot of what happens in my group. So the first point is this: once diabetes has developed, so once the sugar levels in the blood have reached the level where the doctor or nurse is going to diagnose you with diabetes, the horse is out of the barn. And he's probably been running down the road for some time. And the reason I say this is that diabetes results, for the sugar levels to get to the level for diagnosis of diabetes, there has been a very long process of things happening in the background. Diabetes results from chronic progressive disorders that evolve over many years or in fact decades. So the second, and I'm going to tell you how we deal with that in a minute. The second issue is that dietary intake is hard to measure, right? If we do a survey of exposures in uranium mines and lung cancer outcomes, as a researcher, I'm going to have a pretty good idea about what your exposure was to those carcinogenic elements in the mine, right? Some things can be measured better than others because of the properties of that behavior or that exposure. Smoking, for example. We can do a pretty good job characterizing people's lifetime exposure to dose and duration of cigarette smoking. Diet is not so easy, right? Mostly we've been using questionnaires which have problems associated with them. They're very burdensome to participants. A good food frequency questionnaire is 150, 200 items. It takes half an hour to an hour to administer. And people get tired, right? They're volunteering for your studies. They've got other things to do. It's a real burdensome thing. We're getting better at designing questionnaires and methods of administration of questionnaires to overcome some of that, but it's still an issue. Diet is complicated to remember. You know, you can remember certain things very distinctively, but what you ate last week or a year ago is much more of a complex cognitive process. So this reliance on memory, the fact that biases can be introduced and other errors of measurement can be introduced. It makes it difficult. And it's one of the reasons that the public and the news media and the scientists are so frustrated by the unevenness of results on nutrition and health. One of the factors there is that the measurement is very difficult. It's very noisy. So how do we overcome these challenges? Well, the first one, the horses out of the barn problem, the way that my group deals with this is to focus on disorders that occur very early, well before the onset of elevated glucose levels. And in that, excuse me, in that I'm referring to insulin resistance, which is the poor usage of insulin in the body for its normal functions. It's almost like an engine that needs a tune-up. It's working, but it's not working very efficiently. And insulin resistance has been documented now to occur decades prior to the elevation of blood glucose levels. Similarly, the cells in your pancreas that produce insulin, years ago, we thought that those don't start to fail until quite late in the game. Well, it turns out that beta cell dysfunction or poor functioning of the cells that make insulin, this occurs very, very early as well. So my group studies people before the onset of diabetes. We measure these factors and we try to relate them to important exposures and nutrient properties such as dairy product intake. How do we overcome the difficulty of measuring diet? Well, we are moving into the area of biomarkers, things that circulate in the blood, things that you can measure in tissue, that are a reflection of the intake of specific components of the diet. The caveat here is that we don't have very many good biomarkers of dietary intake, but there are a few that are coming to the surface that may be very helpful for us. So my program under the cluster is trying to advance our understanding of the role of dairy products in the prevention of type 2 diabetes through the study of the impact of fatty acids or fats in dairy products on the physiological traits underlying diabetes. So you all know this very well already. Dairy is a wonderfully complex matrix. There are sugars, there are proteins, and there are also fats across a very wide spectrum. We have mono and polyunsaturated fats, and we also have saturated and trans fats, which for many years, and Jean-Philippe alluded to this in his presentation, were the bad guys. The story is changing. As we look more carefully at this evidence, a different story is emerging, and it appears that the saturates and trans that come from dairy products at the very least are neutral, and there may be some benefit to them. Now, the advantage that we have as researchers is many of the fats that we find in dairy products, one of the complicated things about studying fatty acids in the circulation and health is that some of these things are made inside, right? They're endogenously produced. And so palmitic acid comes from a whole bunch of different places. Some of these fatty acids that we consume from dairy are entirely exogenously generated. They come from the outside, so if they're in the circulation, we know that they have come from dairy products, and so they're good biomarkers of whether or not people are consuming dairy and at what level. And I'm listed a few here. I won't go into the scientific names, but the scientists in the room will recognize some of these from the literature. And there's some, put some references here for those of you that are interested. Now, in addition to being biomarkers, there is some increasing evidence that some of these may have bioactive components, and so they may impact some of the traits that I talked about earlier. Again, I'm not going to go into tremendous detail here, but you can see some of the evidence sort of summarized here, and it's quite interesting that some of these fatty acids may have a beneficial role on insulin resistance, which is one of these early disorders that I told you about earlier. All right, and so I'm almost done. I'm just going to spend a couple of slides telling you about some of the research that's occurring in my group. And so a couple of years ago, we published a paper in the American Journal of Clinical Nutrition. This was led by Ingrid Santorin, who is pictured here, and was a master's student in my group at the time. She's now enrolled in the PhD program with me. And Ingrid and I worked on a study called the IRAS study, which is an American study that I first became connected with when I was during my training period as a postdoctoral fellow. And IRAS is an older study, but they have a lot of biospecimens that are saved, and we had the opportunity to go back and measure some of these fatty acids that I told you about, and look at them in relation to the development of diabetes, as well as some of these very important underlying disorders that occur early in the pathogenesis of diabetes. I'm not going to go into detail on this slide, other than to tell you that the advantage of this study is that the detail with which things have been measured was exquisite. In fact, the way research funding is going these days, there probably won't be another study like this because it's just too expensive to do. The main finding, just if you concentrate on the top part of this graph, again, these are odds ratios, which is similar to the relative risk that I told you about earlier. What Ingrid found was that circulating levels of pentadecanoic acid, or 15O, in the blood, higher levels were related to a lower chance of developing diabetes after 5 years. This estimate was relatively stable across important subgroups in the population, men and women, whether people had prediabetes or not, and across the different ethnic groups. So again, well, I can't show you all of this. It was a very complex paper, but 15O did actually track with the consumption of dairy products in this population. It was related to a lower risk of diabetes and better insulin sensitivity and better functioning of the pancreas. What was interesting was that the other sort of exciting fatty acid that's being talked about now in the context of dairy, trans-palmitoleic acid, and some of you will have seen the results from Dr. Mosafarian's work from Boston, we found that this wasn't, in fact, impacting diabetes. And then when we went and looked at how it tracked with foods, because of the age of this cohort, which was collected during a period before the reformulation of trans fats in foods, it was actually picking up an industrial trans fat signal and not a dairy a dairy signal. So last slide, what we're working on right now. So in Toronto and in London, Ontario, we have a study called PROMISE, which is again looking at people who do not yet have diabetes but are otherwise at high risk for diabetes. I'm working with a colleague at the University of Toronto, Richard Bazinet, who is a lipid biochemist, and he has technologies that allow us to look at a very detailed spectrum of the parts of the fat spectrum that come from dairy products. And we're able to measure a wide range of these and look at how they relate to the traits underlying diabetes. Again, this is brand new preliminary data. We have much more analysis to do, but certainly transpalmitoleic, transvasenic, and 15O seem to be beneficially related to a better use of insulin in the body, better insulin sensitivity. And that just summarizes that here. So where do we go from here? Every scientist has their own opinion about the next steps, the next exciting thing we need to do. Well, certainly we need additional data on the roles of dairy subtypes, the question of high-fat dairy in particular, and its components, milk, cheese, yogurt. I believe that we need to understand how dairy impacts some of the early disorders before diabetes occurs, inflammation, insulin resistance, body weight, etc. Dr. Wheeler is going to talk next about body composition, and I think that's going to be very important to hear those findings. We need to work. I'm an epidemiologist. I work very hard to assemble and follow large groups of human participants, but I collaborate with other scientists who can expand. I work with them to expand the work to, in this case with Dr. Bazinet, we've been able to do some measurements of fatty acids that I think are quite innovative. And this is work funded by the cluster. In terms of trials, the trials are getting better at making relevant designs for dose, duration, dairy form, and relevant outcomes. So just to conclude, the observational studies are telling us that total dairy intake is inversely associated with diabetes incidence. We see some inconsistencies regarding dairy subtypes, but certainly the signal is at the very least neutral. Trials have been inconsistent, but there are some design issues that need to be addressed. What are the mechanisms? Well, as a scientist, this is incredibly exciting because I haven't even talked about protein, I haven't talked about some of the other bioactives in dairy, but there are a number of very intriguing, exciting mechanisms that may be at play, including those related to the fatty acid spectrum of dairy. Shelley forced me to revise this slide. I had a whole bunch of names in here. To do science today, you work with a lot of people, a lot of collaborators. That's the way science is done today. I want to acknowledge Richard Bazinet from the University of Toronto, who runs the Fatty Acid Lab, and he's really opened up a whole new area for us with his technologies. I have a lot of trainees in my group. Ingrid Santorin did most of the work I showed today. Science, to get the right answer from science is expensive, and so we're all looking for funding, and I'm supported by a number of agencies, but I want to particularly acknowledge today the Dairy Cluster and the support they've given for the work I showed you today. Thank you very much.
That was great, thank you. Yes, I did do some arm twisting a little on some of those slides. But I find we want all the names there, and then we want the numbers, and I find the numbers also impact as well. Just so that you guys don't see a page full of names sometimes, there are so many collaborators that are working on some of these projects. It's really outstanding. And this is, attributed to the financing that dairy farmers are providing with their partners to some of these projects. So Xstrata gives you the magnitude of the investments and how that reaches across the country into institutions and research centers.
Next speaker, Dr. Chouinard from Laval University. Dr. Chouinard is a professor at the Laval University. Since 2009, he has the Industrial Research Chair on Nutritional Control of the Population, Milk Components in the Cow. Dr. Chouinard will unveil the result of his work. Thank you for being with us.
Donc, bonjour tout le monde.
Good morning, everyone. So as you have been told, I'm going to be outlining the results of a study on production. This is work that is going on in the Animal Sciences Division. We are looking at the fat content of milk and the aromatic components, the prevention of oxidization, and diagnosis of digestive issues. We also have a study on fatty acids that have an impact on health overall. And this is what I'm going to be dealing with now. Omega-3 fatty acids. That's the first part of my presentation. This isn't a new issue. They're already on the market, dairy products enriched in omega-3 fatty acids, milk and butter produced in plants. We add linseed oil to milk in some cases. So these are dairy drinks. There are also other grocery products available. And we changed the food components in the diet of the cow, and this has an impact on human nutrition. These products have been on the market for about 10 years, so there's some interest. It's not a high volume of production, but there is some interest for this. When you want to enrich a product with a given nutrient, often you talk about the recommended servings per day, what is deemed sufficient to consume. Mistakes can be made because sometimes the bar is set too high. And people, when we modify a food stuff, think that once the work is done, with one portion you'll succeed in having the recommended daily dose, like taking a pill or a capsule. But we don't just consume one food stuff during the course of a day. So we have to rely on labeling to know exactly how much is being consumed. There are Canadian rules and regulations in this area. And you can't just make an empty allegation about a product to say that it's a source of omega-3s. What the law says is that the product must contain 0.3 grams per portion in order to have a label that says that it offers omega-3 fatty acids. So I took a glass of milk, whole milk for my illustration, 8 grams of fat. So a whole glass of milk contains 8 grams of fat, 3.7 of omega-3s. That is the target in order to be able to put on the label that it's omega-3 enriched. Why is it so difficult to achieve this through the food given to the cow? Well, it's because the microorganisms in the cow's rumen don't like fatty acids. They're there to digest fiber, and the presence of these fatty acids is an issue, so they've developed a mechanism to eliminate them. And this process is called biohydrogenation. You find bacteria even in forage. And this is the process that transforms these acids, and that's why you don't find many saturated fat. So what do we do? A lot of issues have been studied. Heat processing, coatings of some grains, etc. And we looked at calcium salts in particular. Here you've got alpha-linoleic acid that you find in linseed and forages, and if you attach those two molecules to a calcium atom, well, you prevent the cow's system from experiencing the biohydrogenation process. What we noted is that as the foodstuffs go through the stomach of the cow, the chain can be broken. Calcium salts, producers use them. With Megalac, for example, these are calcium salts prepared with palm oil. So these fatty acids are saturated, and this is an energy supplement given to cows. We can also use other oils like linseed to produce these calcium salts. This has already been tested. Their effectiveness is limited, but some companies did market this type of product. When we look at commercial products obtained with an industrial calcium salt production process, when I was a student, I produced them myself in a lab, but now there are companies doing this. When we look at these products, we see that the industrial process for producing calcium salts leads to a mixture of different particles of different sizes. So we sifted them and we obtained smaller and larger fractions. And our hypothesis is that calcium salts are imprisoned in a particle. They'll be protected physically as they go through the rumen. So there is physical protection for large particles. If the particle leaves the rumen before it's attacked by bacteria, there will be a better transfer of the omega-3s from the nutrition of the cow to her milk. This is what we saw in our work in the research chair. So we used linseed here as a comparator. It wasn't protected, and we offered 2 cows calcium salts. So the fine salts, we wanted to run trials with both fine salts and coarse salts, and here you have the content in omega-3 fatty acids. So here we began at 0.6% with our oil. We doubled this using the fine salts, so it is useful to use calcium salts, and when we use the coarse salts, we doubled this yet again. So we achieved 4 times the concentration with the coarse salts. When you look through the literature, and I've been working on this for many years now, and this is a result that we never were able to achieve. It's never been equaled in literature for a transfer of omega-3 fatty acids without using chemicals to protect or to bypass the rumen artificially. So this is more natural, and this is the first time that we've achieved such results. In order to compare this with the products available on the market, we investigated. We went to a store and we bought the various dairy products available to make a comparison with our products to see how they could position themselves. So we started with regular milk, bought off the shelf, whole milk, just regular milk. We could do the same calculations with 1%, 2% milk. The more you remove fat from the milk, the less you'll have fatty acids in the product. So we used regular milk, and this contributes about 40 milligrams of omega-3s in a glass of milk. There are other products available on the market. DHA milk, it's C22-6. These products are obtained by adding oil or fish meal in cows' rations, because in fish flesh there are omega-3s. So the cow absorbs this, and some of it is found in the milk, 20 milligrams more. In Ontario, you've got Dairy O. In Quebec, you have Smart Milk, which is produced in this way. On the market, there are also organic milk products. Organic milk, well, when you buy organic whole milk, and this is what we used for our study that was very comprehensive, it's standardized at 3.8% fat instead of 3.25. So right away, right there, even if you don't change anything else, there's more fat in one portion. And in organic farms, cows get more forage and they're pastured out in the summer. So all of these elements increase the omega-3. So it gives you about 100 milligrams of omega-3s in a portion of organic milk compared to conventional, regular milk. And it's not because it's organic, but it's because of the feeding practices on organic farms for cows. The other product we find on the market, milk beverage products. For that category, you take regular milk, it's skimmed, you make butter and butter oil, and linseed is added to it, and all of this is emulsified in the drink, and you therefore have a milk beverage that contains exactly what's prescribed for nutritional purposes, 33 milligrams, or rather 300 milligrams. And if you have 1% milk, you'll have your 300 milligrams. It'll just be more concentrated. And the same dose will apply for 2% milk. Finally, our experimental milk with the dose we had earlier, expressed in grams of fat. If we standardize it at 3.25, it gives us 350 milligrams of fatty acids. So we're close to the standard. We haven't achieved it yet, but it's a quantity of omega-3s that is considerable. It's higher if we compare this with what is deemed sufficient for alpha-linolenic acid and the 18-3. It's 1 gram per day for women that's recommended. If a glass of milk provides 250 milligrams, you would achieve the necessary daily intake levels with 1 glass. With other products, you'd have to drink 25 glasses of the beverage or of the milk to achieve that level. Second part of my presentation, branched chain fatty acids. We talked a lot about fatty acids today. We find this in milk. Fatty acids, normally, the most frequent one, has a carboxyl chain. This is why we call it an acid. Here we have the branched chain fatty acid that we studied. And if you give palm oil to your cows, this increases this type of fatty acid. Here we have a linear chain with additional carbon. If the carbon, or we're looking at the branched chain, it's iso if it's in the first line and anti-iso in the second line. Why is this interesting to us? Well, we're seeing impacts on health with these fatty acids. An important source of fatty acids, branched, is vernix. Vernix is a viscous substance that's on baby's skin. You see it at birth, and it protects the skin of the baby during gestation in the amniotic fluid. We see it here. There's a great diversity of these fatty acids. It's very abundant, produced by the skin of a baby, a newborn. During gestation, there are particles of this that flow into the amniotic fluid, and the baby, during the first term of gestation, drinks amniotic fluid. So some researchers said that it could be a source of nutrition for babies, and they assessed the presence of these fatty acids in the meconium, which is the first bowel movement of the baby at birth. And we found this in the meconium, which proves that it's present in amniotic fluid. Some of this is absorbed because some of it disappears. Premature babies don't have the opportunity to consume amniotic fluid. And this can lead to the development of certain problems with their digestive system. There's a problem with premature babies that we often see. It's necrotizing enterocolitis, which is a degeneration of the intestinal tissue. Here you've got some slides. With this disintegration, the intestinal tissue disintegrates, and this often leads to the death of premature babies. We wanted to know if there was a link with the presence of fatty acids. At Cornell, this was studied by some researchers there. So by cesarean, they studied premature rats, and they fed them with a lactated mix without any branched chain fatty acids, and another group with branched chain fatty acids. And we saw this necrotizing enterocolitis incidence. It was 50%, and it was slashed in half for those baby rats who had received the source of branched chain fatty acids. And there was the comparison group that got their mother's milk, and they didn't suffer from this. So the mother supplied this to her baby with all the other nutrients in her milk. So here we see a link with a mother with a premature baby. If she has a diet rich in these fatty acids, she would give more to her baby through her milk. So now the the mode of action, they're going to promote the growth of certain bacteria. So here we have the abundance of Bacillus subtilis, which is a probiotic. The presence of this bacteria in one's intestine increases intestinal health. And we see that when the subjects suffered from necrotizing enterocolitis in their intestine, there was a drop in this bacteria. So we wanted to determine if branched-chain fatty acids increase the presence of this bacteria, its abundance. And they studied the diet of those subjects that had fatty acids added. So there would be a reaction. At birth, it would act like a probiotic. You'd colonize intestines in this way, and this would enhance fatty acid growth. To conclude, our work was based on production, and we saw that there could be a link between the mother's diet. If she drinks milk with branched-chain fatty acids, this could find its way to her own milk that she would give her baby if she's nursing. And there are quantities of these fatty acids in mother's milk, and we wanted to determine the impact. So we compared cows that got forage, pasture, and silage. We wanted to compare the impact on the aromatic qualities of milk and also the fatty acid content. And what we saw is that for various branched-chain fatty acids, there's always a lower quantity, significantly lower, when cows are fed silage as opposed to hay. For some fatty acids, it's higher, again, when the cows are pastured. So we'd have to study this in global rations. Two other results: the effects of feed rich in concentrates. You can give different types of concentrates to cows. Here you had cows with a 30% or 70% concentration ration. When there's a lot of grain in the ration, a lot of concentrates, a lot of the fatty acids find themselves in the milk. There are bacteria that digest the fiber in the rumen for cows that consume a lot of fiber, and there are other bacteria that digest the concentrates. So we have to study these mechanisms further. Here we're looking at the effects of linseed oil, because we're using linseed oil to increase the omega-3 in the milk. And when we add linseed oil, about 3% in the cow's rations, all of the branched-chain fatty acids dropped in the same proportion because the cow integrates this in her milk. And if we enrich it in omega-3, we lose the branched-chain fatty acids, because what you get on one side, you lose on the other. In conclusion, the first part of my presentation dealt with the production of milk enriched with omega-3 fatty acids. And we saw that we needed certain levels in order to mention enriched on the label. It may not apply to your situation. Perhaps you don't want to market omega-3 enriched milk, but what you must understand is that if omega-3 fatty acids are good for the cow, they're good for us. If we increase omega-3 fatty acids that bypass the rumen and they find themselves in the bloodstream, yes, there'll be more in the milk, and in passing, the cow too can benefit from additional omega-3 in her feed. So any strategy that would allow us to increase the omega-3 in the milk would increase omega-3s for the cow, and there might be a positive impact on her health and her reproductive health as well. With regard to branched-chain fatty acids, we saw potential effects for health. We're starting to see results in that area. We'll have to look at this further to see what happens for adults. It's a whole new focus for research that should be looked into. To conclude, I don't have statistics as to the participation in our study. Our work is nearly finished now at the chair, and it allowed us to recruit new researchers, Edith Charbonneau and Rachel Gervais. And this was a wonderful addition to our animal sciences department. We achieved results short term, but these individuals are going to stay in the future, so there will be very positive results in the area of human resources and others for the chair, the research chair. So I'd like to thank our partners. DFC was a partner for us. It also benefited from support by NSERC, Novalet, Valacta, and the Deschambault Animal Science Center. Thank you for your attention.